Welcome to deep science sleep. Close your eyes and let the world outside fade away. Feel the weight of your body sinking into the soft embrace of your bed, each muscle unwinding, releasing the tensions collected throughout the day. Your breath becomes deeper, slower, more rhythmic, like gentle waves lapping at a moonlit shore. The day's thoughts begin to dissolve, their sharp edges softening, blurring, drifting away like clouds dispersing across an evening sky. You are safe here, wrapped in the quiet darkness, protected from the demands of waking life. Nothing requires your attention now. Nothing needs to be solved, accomplished, or understood. This is your time to rest, to retreat inward, to let consciousness slip gently away. As you float in this liminal space between waking and sleeping, your mind begins its nightly journey into landscapes you cannot predict, stories you did not write, and experiences that will feel completely real until morning light filters through your windows once again. Welcome to the world of dreams, where science meets mystery, where your brain becomes both artist and audience, creating elaborate scenarios for reasons scientists are only beginning to understand. The mystery that haunted ancient civilizations. For as long as human beings have possessed the capacity for wonder, dreams have occupied a sacred space in our consciousness. Long before the first scientific instrument recorded the electrical patterns of a sleeping brain, our ancestors gazed at the night sky and pondered the strange visions that visited them in darkness. The enigma of dreaming transcends culture, geography, and time itself. In every corner of the ancient world, from the ziggurats of Mesopotamia to the temples of the Nile Valley, from the mountain monasteries of Tibet to the rainforests of the Amazon, human societies developed elaborate theories about the origin and meaning of these nocturnal experiences. The ancient Egyptians, masters of both mathematics and mysticism, believed dreams were messages from the divine realm. They created dream books, intricate papyrus scrolls that cataloged hundreds of dream scenarios alongside their purported meanings. These weren't simple superstitions scrawled hastily in the margins of history. Rather, they represented sophisticated attempts to systematize the seemingly chaotic realm of dream content. Egyptian priests, who served simultaneously as healers, astronomers, and interpreters of the sacred, would be consulted when someone experienced a particularly vivid or troubling dream. In the temples of Serapis, a Greco-Egyptian deity, people would engage in a practice called incubation, deliberately sleeping in sacred spaces to receive healing dreams or prophetic visions. They believed that in sleep, the boundary between the mortal and immortal worlds grew thin, permeable, allowing communication between realms that remained stubbornly separate during waking hours. The Mesopotamians shared this reverence for dreams but added their own unique interpretations. In the Epic of Gilgamesh, one of humanity's oldest surviving works of literature, dreams serve as crucial plot devices, offering warnings, predictions, and divine guidance. The hero Gilgamesh receives prophetic dreams about his companion Enkidu, visions that foreshadow their adventures and ultimate fates. These weren't mere literary flourishes. They reflected a deeply held belief that dreams possessed agency, that they were dispatched by gods who wished to communicate with mortals. Assyrian kings would consult dream interpreters before military campaigns, believing that the visions of sleep could reveal strategic advantages or warn of impending defeat. Clay tablets discovered in ancient Mesopotamian cities contain detailed records of royal dreams and their interpretations, creating an archaeological record of how seriously these cultures took their nocturnal visions. Classical Greece and Rome inherited this fascination but began to approach dreams with a more philosophical, less purely religious perspective. The great Greek physician Hippocrates, often called the father of medicine, proposed that dreams might reflect the body's physical state rather than messages from Mount Olympus. He suggested that certain dream content could indicate illness or imbalance in the humors, those bodily fluids that ancient medicine believed governed health. A person dreaming of floods might have excess phlegm, while dreams of fire could indicate too much yellow bile. This represented a radical shift in thinking, moving dreams from the realm of the purely supernatural into the domain of natural philosophy, the precursor to modern science. Aristotle, that tireless categorizer of natural phenomena, wrote extensively about dreams in his work on dreams and on divination in sleep. He rejected the notion that dreams were divine communications, arguing instead that they resulted from sensory impressions that persisted even after the external stimuli had ceased. When you dream of sounds, Aristotle proposed, you're experiencing echoes of sensory data collected during wakefulness. He recognized that dreams occur during sleep, a state he viewed as a kind of partial consciousness, and he noticed that external stimuli could influence dream content. A loud noise in the environment might be incorporated into a dream, transformed by the sleeping mind into something that fit the dream's narrative. Aristotle was grasping toward a materialist understanding of dreams, one that would take more than 2,000 years to develop fully. The Romans contributed their own variations on dream interpretation. Artemidorus of Daldis, who lived in the 2nd century AD, compiled the Oneirocritica, a 5-volume treatise on dream interpretation that remained influential well into the Middle Ages. Unlike earlier dream books, Artemidorus attempted to create a more systematic approach, categorizing dreams by type and considering the dreamer's personal circumstances when interpreting dream symbols. He recognized that the same dream image might mean different things to different people, an insight that would resonate with later psychological approaches to dream analysis. In the Far East, dreams occupied different but equally significant positions in philosophical and religious thought. Traditional Chinese medicine viewed dreams as intimately connected to the body's energy flows and organ systems. Different types of dreams indicated imbalances in different organ networks. Excessive dreaming itself was considered a sign of disharmony, suggesting that the shun, the spirit housed in the heart, was restless and unable to settle peacefully during sleep. Taoist philosophy developed elaborate practices around dream cultivation, techniques for achieving greater awareness and control within dreams, practices that would later be recognized as early forms of lucid dreaming. In India, dreams were incorporated into both Hindu and Buddhist philosophical frameworks. The Upanishads, ancient Sanskrit texts, describe dreams as one of several states of consciousness, existing on a continuum between waking and deep dreamless sleep. The Mandukya Upanishad specifically identified 4 states of consciousness, with the dream state representing a middle ground where the soul experiences subtle impressions and karmic traces. Buddhist texts developed similar frameworks, viewing dreams as manifestations of the mind's fundamental nature, neither entirely real nor entirely unreal, perfect examples of the illusory quality of all phenomena. Indigenous cultures around the world developed their own rich traditions of dream interpretation and use. Among many Native American tribes, Dreams were viewed as sources of personal power and spiritual guidance. Vision quests, ceremonial practices involving fasting and isolation, were designed to induce powerful dreams that would provide direction for major life decisions. The Iroquois people of northeastern North America believed that dreams revealed hidden desires of the soul, and they developed practices for acting out dreams ceremonially to satisfy these deep psychological needs, a practice that prefigures modern psychotherapy in intriguing ways. Australian Aboriginal peoples incorporated dreams into their understanding of the dream time, a foundational concept in their cosmology. The dream time refers both to the ancestral era when the world was created and to the ongoing spiritual reality that can be accessed through dreams and ceremonies. In this framework, dreams aren't separate from waking reality but represent different modes of experiencing the same fundamental truth. The boundaries between past, present, and future, between the individual and the collective, between the physical and spiritual, all become permeable in dream consciousness. The Sainwa people of Malaysia developed perhaps the most sophisticated pre-modern techniques for working with dreams. Anthropological accounts, though later questioned in their details, described a culture where children were taught from an early age to control their dreams, to confront threatening dream figures, and to extract useful knowledge from dream experiences. Whether or not these accounts were entirely accurate, they captured imaginations in the Western world during the 20th century, inspiring various movements around lucid dreaming and conscious dream work. What unites all these diverse cultural approaches is a fundamental recognition that dreams matter, that they're worth attending to, interpreting, and learning from. Ancient peoples may not have understood the neurobiological mechanisms underlying dreams, but they recognized that dreams reflected something important about human psychology, spirituality, and perhaps even health. They noticed patterns in dream content, connections between waking experiences and dream scenarios, and the powerful emotional impact that dreams could have on waking consciousness. Yet, for all this attention, for all the dream books and temple practices and philosophical treatises, the fundamental question remained unanswered. Why do we dream? What possible purpose could these often bizarre, sometimes frightening, occasionally prophetic-seeming experiences serve? The ancients offered theological, philosophical, and proto-medical explanations, but they lacked the tools to peer inside the living, dreaming brain, to measure its electrical activity, to observe which regions activated during different sleep stages, to understand the biochemical changes that accompany the shift from waking to sleeping consciousness. That technological capability would have to wait. For thousands of years, dreams remained mysterious, their origins unknowable, their purposes debatable. Philosophers could theorize, priests could interpret, and individuals could wonder, but no one could definitively answer the most basic questions about these nightly mental adventures. The sleeping brain kept its secrets, locked away behind closed eyelids, protected by the impenetrable barrier of unconsciousness. Science, when it finally turned its attention to dreams, would need to develop entirely new tools, new methodologies, new frameworks for understanding consciousness itself before it could begin to penetrate the mystery that had fascinated humanity since the dawn of self-awareness. The brain's hidden architecture. To understand why we dream, we must first explore the remarkable architecture of the sleeping brain, a landscape as complex and intricate as any structure humanity has ever built. The human brain contains approximately 86 billion neurons, each one a sophisticated biological machine capable of receiving, processing, and transmitting electrical and chemical signals. These neurons don't work in isolation. Each one forms thousands of connections with other neurons, creating a network of staggering complexity. If you attempted to count every synaptic connection in a human brain, one connection per second, you would need more than 6 million years to complete the task. This vast neural network generates consciousness, processes sensory information, controls movement, stores memories, regulates emotions, and produces the ephemeral, often bizarre experiences we call dreams. The brain is not a uniform organ. Rather, it's composed of distinct regions and structures, each with specialized functions that contribute to our mental life. Understanding these regions and their interactions during sleep is crucial for comprehending how and why dreams emerge. Let's begin our tour in the deepest, most ancient parts of the brain, structures we share with our reptilian ancestors, and work our way outward to the regions that make us distinctively human. Deep within the skull, nestled at the base of the brain where it connects to the spinal cord, lies the brainstem. This thumb-sized structure controls many of our most vital functions: breathing, heart rate, blood pressure, and the sleep-wake cycle itself. Within the brainstem, a region called the pons plays an absolutely critical role in generating the type of sleep most associated with vivid dreaming. The pons contains clusters of neurons that become remarkably active during rapid eye movement sleep, the stage where dreams are most intense and memorable. These neurons send signals throughout the brain, initiating a cascade of changes that create the unique neurobiological state we experience as dreaming. One particular group of cells in the pons deserves special attention: the cholinergic neurons of the pedunculopontine tegmentum and the laterodorsal tegmentum. These tongue-twisting names refer to specific neural populations that release acetylcholine, a neurotransmitter crucial for generating REM sleep and its associated dream experiences. During waking hours, these neurons are moderately active, helping to maintain alertness and attention. But when we enter REM sleep, their activity surges dramatically, flooding other brain regions with acetylcholine. This neurochemical shift helps explain many of the distinctive features of dreams: their hallucinatory quality, their emotional intensity, and the way they can feel so vividly real while simultaneously being utterly bizarre. Just above the brainstem sits the thalamus, sometimes called the brain's relay station. Nearly all sensory information flowing into the brain, from vision and hearing to touch and pain, passes through the thalamus before reaching the cerebral cortex, where conscious perception occurs. During waking hours, the thalamus actively gates this sensory information, filtering out irrelevant stimuli and directing important signals to appropriate cortical regions. But during sleep, and particularly during REM sleep, something fascinating happens. The thalamus substantially reduces its transmission of external sensory information. The sleeping brain becomes disconnected from the outside world, no longer receiving or processing the sights, sounds, and sensations that constantly bombard us during waking consciousness. Yet the thalamus doesn't shut down entirely. Instead, it continues to send signals to the cortex, but these signals now originate internally, from other brain structures rather than from sensory organs. This internal activity, disconnected from external reality, forms part of the neurobiological basis for dream experiences. Adjacent to the thalamus lies the hypothalamus, a small but extraordinarily important structure that regulates hunger, thirst, body temperature, and crucially for our purposes, the sleep-wake cycle. Within the hypothalamus, certain groups of neurons act as master switches, controlling transitions between sleep and wakefulness. The lateral hypothalamus contains neurons that produce orexin, also called hypocretin, a neurotransmitter essential for maintaining wakefulness. When orexin-producing neurons are active, we feel alert and awake. When they fall silent, as they do during sleep, consciousness fades. People with narcolepsy, a sleep disorder characterized by sudden, uncontrollable sleep attacks and vivid, often frightening dream experiences intruding into wakefulness, typically have lost most of their orexin-producing neurons. This tragic neurological condition provides powerful evidence for orexin's crucial role in regulating the boundaries between sleep and waking, between dreaming and waking consciousness. Moving upward and inward, we encounter the limbic system, a collection of structures involved in emotion, memory, and motivation. The limbic system includes the amygdala, hippocampus, and cingulate cortex, among others. The amygdala, a small, almond-shaped structure, serves as the brain's emotional processing center, particularly for fear, anxiety, and other negative emotions. During REM sleep, the amygdala becomes highly active, even more active than during waking hours. This hyperactivity helps explain why dreams so often contain emotional content, why they can be frightening or anxiety-provoking, and why we sometimes wake from dreams with our hearts racing and palms sweating. The amygdala is processing emotions during sleep, possibly helping us work through emotional experiences and develop appropriate responses to threatening situations, even hypothetical ones that exist only in dreams. The hippocampus, another limbic structure, is crucial for forming new memories and for spatial navigation. This seahorse-shaped region takes short-term memories, the transient impressions of daily experience, and helps consolidate them into long-term storage in the cortex. During sleep, particularly during the deep slow-wave sleep that precedes REM periods, the hippocampus replays neural patterns established during waking hours. Researchers recording from individual neurons in the hippocampus of rats have observed that sequences of neural firing that occurred while the rats explored a maze are replayed, often in accelerated form, during subsequent sleep. This neural replay appears to be crucial for memory consolidation, for transforming fleeting experiences into lasting memories. The hippocampus's activity during sleep may also contribute to dream content, as recent experiences are reprocessed and integrated into existing memory networks. Finally, we reach the cerebral cortex, the wrinkled outer layer of the brain that makes us distinctively human. The cortex is where higher-order thinking occurs. Reasoning, planning, language, voluntary movement, and conscious perception. Different regions of the cortex specialize in different functions. The occipital cortex at the back of the brain processes visual information. The temporal cortex on the sides handles auditory processing and certain aspects of memory. The parietal cortex at the top integrates sensory information and supports spatial reasoning. And the frontal cortex, particularly the prefrontal regions at the very front of the brain, is involved in executive functions like planning, decision-making, and controlling impulses. During REM sleep, activity in different cortical regions varies dramatically. The visual cortex becomes highly active, even though our eyes are closed and no visual information is entering the brain. This internal activation of visual processing areas creates the rich, often spectacular visual imagery of dreams. We see people, places, and things that don't exist in the external environment, generated entirely by the brain's internal activity. Meanwhile, the prefrontal cortex, that seat of rational thought and executive control, becomes relatively quiet during REM sleep. This deactivation helps explain several peculiar features of dreams. Why we accept bizarre scenarios as perfectly normal while dreaming, why we lack insight that we're dreaming until we wake up, why logic and sequential reasoning often seem absent from dream narratives, and why we have such poor control over dream content compared to our thoughts during waking. The deactivation of the prefrontal cortex during dreaming is particularly significant. This region is responsible for reality monitoring, for distinguishing between imagination and perception, between internal mental events and external reality. When it becomes less active during REM sleep, this reality monitoring capacity diminishes, and we lose the ability to recognize that our current experiences are internally generated rather than externally real. We're essentially experiencing a temporary, reversible form of psychosis, a state where internal mental events are confused with external reality, where logical impossibilities are accepted without question, where our own identity might shift or fragment. Other cortical regions show interesting patterns during REM sleep as well. The medial prefrontal cortex and the anterior cingulate cortex, both involved in emotional processing and self-referential thinking, remain active during REM sleep. Their activity may contribute to the sense of self that persists in dreams, the feeling that I am experiencing these events, even when the dream self behaves in ways quite different from our waking personality. The inferior parietal cortex, involved in spatial processing and navigation, also shows activation during REM sleep, which may help explain why dreams often involve movement through space, travel from place to place, even when those places violate the laws of physics or seamlessly blend impossible locations. The interaction between these various brain regions creates the neurobiological substrate for dreaming. It's not any single structure that generates dreams, but rather the specific pattern of activation and deactivation across multiple brain areas. The brainstem initiates REM sleep and triggers the cascade of neurochemical changes. The thalamus gates sensory information while continuing to send signals to the cortex. The amygdala processes emotions intensely. The hippocampus replays recent experiences. The visual cortex generates imagery. The prefrontal cortex loses its supervisory control. And the cortex as a whole weaves these various signals into a narrative experience, creating the coherent feeling stories we remember as dreams. But brain structures alone don't tell the whole story. The sleeping brain is also bathed in a dramatically different neurochemical environment compared to waking hours. Neurotransmitters, those chemical messengers that allow neurons to communicate across synapses, show striking changes in concentration during different sleep stages. Understanding these chemical shifts is crucial for understanding why dreams have their distinctive character, why they feel different from waking thought, and why they may serve important functions for brain health and emotional regulation. The REM revolution. In the early 1950s, the study of sleep was about to undergo a transformation as profound as any in the history of neuroscience. The prevailing view held that sleep was a passive state, a time when the brain simply powered down, resting and recuperating from the demands of waking life. People recognized that some sleep was deeper than others, that dreams seemed to occur at certain times, but the notion that sleep itself might be an active, dynamic process with distinct stages, each serving different functions, had not yet taken hold. That was about to change thanks to the stubborn persistence of a graduate student and the wisdom of his mentor, both working in a small laboratory at the University of Chicago. Eugene Aserinsky was not the most obvious candidate to revolutionize sleep science. He had arrived at the University of Chicago in 1950 to begin graduate work in physiology, somewhat reluctantly selecting Nathaniel Kleitman as his advisor. Kleitman, born in what is now Moldova in 1895, had fled revolutionary Russia and eventually made his way to the United States, where he became fascinated by the mystery of sleep. He had published the definitive text on sleep research in 1939, Sleep and Wakefulness, a comprehensive review of everything known about sleep up to that point. By the time Aserinsky arrived in his laboratory, Kleitman was the unquestioned dean of sleep research, though the field itself was small and somewhat marginalized within mainstream physiology. Kleitman assigned Aserinsky what seemed like a relatively mundane task: observe the blinking rates of sleeping infants. The hypothesis was that blinking patterns might reveal something about the depth of sleep. Aserinsky spent months watching infants sleep, a tedious process that yielded little of interest. Frustrated with his lack of progress, and perhaps sensing that infant sleep research wasn't going to make his career, Aserinsky decided to shift his focus to adults. He had noticed that sleeping infants sometimes showed periods of eye movement beneath their closed lids, and he wondered if similar movements occurred in adult sleep. To investigate this question, Aserinsky needed a way to record eye movements objectively without relying on direct observation, which was impractical for studying sleep throughout the night. He hit upon the idea of using the corneoretinal potential, a naturally occurring electrical voltage difference between the front and back of the eye. By placing electrodes near the eyes, he could detect when the eyes moved, as these movements would create small electrical signals. Combining this with EEG recordings of brain activity, Aserinsky could monitor both eye movements and brain states throughout the night. What he discovered on that fateful night in late 1951 would change sleep science forever. Aserinsky's first all-night recording subject was his 8-year-old son, Armand. As the boy slept, the recording equipment traced out the expected patterns of slower, rolling eye movements that had been previously observed during sleep onset. But then, approximately 90 minutes into sleep, something unexpected occurred. The pen's tracking eye movements suddenly began jerking wildly across the paper, recording rapid, darting eye movements unlike anything Aserinsky had seen before. His initial thought was equipment malfunction. He checked the connections, adjusted the settings, but the rapid eye movements continued, occurring in periodic bursts. Aserinsky was witnessing rapid eye movement sleep, though he didn't initially call it that. His first inclination was to name these periods jerky eye movement or JEM periods, a term that fortunately didn't stick. These periods lasted about 20 minutes at a time and were accompanied by distinctive changes in brain wave patterns. The EEG showed a low-voltage, mixed-frequency pattern quite different from the high-amplitude slow waves that characterize deep sleep. In fact, the brain wave patterns during these rapid eye movement periods looked remarkably similar to those of waking consciousness, leading researchers to later call this stage paradoxical sleep, a term still used in some scientific literature. Here was the brain, ostensibly asleep and disconnected from the external world, yet showing electrical activity patterns that resembled wakefulness. Kleitman, ever the skeptic and scientist, demanded rigorous proof before he would accept that this represented a genuine new discovery. He insisted on multiple confirmations, careful controls, and systematic documentation. Together, Aserinsky and Kleitman conducted extensive studies, carefully documenting the characteristics of these rapid eye movement periods. They found that REM periods occurred cyclically throughout the night, appearing roughly every 90 minutes, alternating with periods of quieter, deeper sleep. They noted that the rapid eye movements were conjugate, meaning both eyes moved together in the same direction, suggesting these were genuine eye movements rather than random muscle twitches. They measured changes in breathing rate, heart rate, and other physiological parameters, discovering that REM periods were accompanied by irregularities in these autonomic functions. Most significantly, Aserinsky and Kleitman tested whether these rapid eye movement periods had any relationship to dreaming, that elusive mental phenomenon that people had puzzled over for millennia. They began waking subjects during different stages of sleep and asking them to report any mental content they could remember. The results were striking and unambiguous. When awakened during rapid eye movement periods, subjects reported vivid, detailed, often bizarre dream experiences more than 80% of the time. When awakened during periods of quiet sleep without rapid eye movements, dream reports were much less frequent, and the mental content described was typically less vivid, less narrative, more thought-like than dreamlike. In September of 1953, Aserinsky and Kleitman published their findings in the journal Science in a brief paper titled Regularly Occurring Periods of Eye Motility and Concomitant Phenomena During Sleep. The paper was just 2 pages long with little fanfare, yet it represented one of the most significant discoveries in 20th century neuroscience. For the first time, researchers had an objective marker for dream sleep, a way to identify when someone was likely dreaming without relying on their subjective reports. The discovery opened the door to systematic, scientific investigation of dreams, transforming them from subjects of philosophical speculation into legitimate objects of empirical research. William Dement, then a medical student who would later become one of the world's leading sleep researchers, joined Kleitman's laboratory in 1952 and helped extend Aserinsky's initial findings. Dement coined the term rapid eye movement or REM sleep, and he's often credited with naming the phenomenon, though the origin of the term is somewhat complex, as these historical attributions often are. Dement conducted extensive studies characterizing REM sleep in greater detail, documenting the 90-minute cycle that came to be known as the ultradian rhythm, describing how REM periods lengthened as the night progressed, and demonstrating that REM sleep was a universal feature of normal human sleep, not some anomaly or artifact. The discovery of REM sleep transformed our understanding of sleep architecture. Sleep was no longer viewed as a unitary state, a simple absence of waking consciousness. Instead, researchers now recognize that sleep consisted of distinct stages, each with characteristic brain wave patterns, physiological changes, and potentially different functions. Beyond REM sleep, researchers identified 4 stages of non-REM sleep, stages characterized by progressively deeper sleep and slower, higher amplitude brain waves. Stage 1 non-REM sleep represents the transition from wakefulness to sleep. Brain waves slow from the beta and alpha rhythms of waking to the theta rhythms of light sleep. This stage is brief, typically lasting only a few minutes at sleep onset. Stage 2 is characterized by the appearance of sleep spindles, brief bursts of rhythmic brain wave activity, and K-complexes, sharp, high-amplitude waves that may help protect sleep from external disturbances. Stage 2 comprises the largest portion of total sleep time in adults. Stages 3 and 4, often combined and called slow-wave sleep or deep sleep, show the highest amplitude, slowest brain waves called delta waves. This is the deepest, most restorative sleep, the stage from which it's hardest to awaken someone. These sleep stages don't occur randomly, but rather cycle in a predictable pattern throughout the night. A complete sleep cycle, progressing from stage 1 through deep sleep and into REM, takes approximately 90 minutes. As the night progresses, the composition of these cycles changes. Early in the night, slow-wave sleep dominates, with relatively brief REM periods. As morning approaches, slow-wave sleep diminishes, and REM periods become longer and more intense. This changing architecture reflects different processes occurring at different times of night, with different types of sleep serving different functions. The discovery of REM sleep also revealed that dreaming was far more common than previously recognized. People who claimed they rarely or never dreamed turned out to be experiencing amnesia for their dreams rather than an absence of dreaming itself. When awakened during REM sleep in laboratory studies, even these supposed non-dreamers would report vivid dream experiences. This suggested that everyone dreams multiple times every night, typically 4 or 5 separate REM periods, totaling about 2 hours of dreaming over an 8-hour sleep period. Most of these dreams are simply forgotten upon waking, their content dissipating like morning fog in sunlight. The characteristics of REM sleep began to make sense in light of its association with dreaming. The rapid eye movements themselves, those darting movements that gave the sleep stage its name, may reflect the dreamer's visual scanning of the dream scene. Though this interpretation remains somewhat controversial, studies of people with REM sleep behavior disorder, who act out their dreams due to a loss of normal muscle paralysis during REM sleep, suggest some correspondence between eye movements and dream content. The muscle paralysis itself, called REM atonia, serves a crucial protective function, preventing us from physically acting out our dreams and potentially injuring ourselves or bed partners. In REM sleep, all the major muscle groups of the body become temporarily paralyzed, with the exceptions of the diaphragm and other muscles necessary for breathing, and the extraocular muscles that move the eyes. The neurochemical signature of REM sleep also helps explain the distinctive quality of dreams. During REM sleep, levels of acetylcholine surge dramatically, while levels of monoaminergic neurotransmitters, serotonin, norepinephrine, and dopamine drop to near zero. This unique neurochemical state creates a brain that's highly active yet fundamentally different from the waking brain. The absence of norepinephrine may contribute to dream amnesia, as this neurotransmitter is important for memory consolidation. The high acetylcholine levels support the vivid imagery and emotional intensity of dreams. The combination creates a conscious experience unlike either waking thought or the mental blankness of deep sleep. The discovery of REM sleep raised as many questions as it answered. Why does REM sleep exist? What function does it serve? Why do we need to dream? Are dreams just random byproducts of the neurobiological changes during REM sleep, or do they serve some adaptive purpose? These questions would drive decades of subsequent research, spawning multiple theories and generating insights that continue to shape our understanding of consciousness, memory, and emotion. Theories of why we dream. Armed with the knowledge that REM sleep could be objectively identified and studied, researchers began proposing theories about why dreaming might exist, what evolutionary or psychological purpose it might serve. These theories span a remarkable range, from the purely mechanistic to the deeply psychological, from the firmly empirical to the frankly speculative. No single theory has achieved complete consensus among scientists, and it's entirely possible that dreams serve multiple functions simultaneously, their utility not reducible to any single explanation. Let's explore the major theories that have shaped scientific thinking about dreams, examining the evidence supporting each and considering how they might fit together into a more comprehensive understanding. The activation synthesis hypothesis, proposed by Harvard psychiatrists Alan Hobson and Robert McCarley in 1977, represented one of the first major attempts to explain dreaming in purely neurobiological terms. Hobson and McCarley observed that during REM sleep, the pons generates random electrical activity that propagates throughout the brain, particularly to cortical regions involved in vision, motion, and emotion. They proposed that dreams are the cortex's attempt to make sense of these random signals, to synthesize them into something coherent. In this view, dreams have no inherent meaning or purpose. They're simply the mind's effort to create a narrative from neural noise, much like seeing shapes in clouds or faces in random patterns. The activation synthesis hypothesis was revolutionary and controversial. It challenged prevailing psychoanalytic views that dreams were meaningful expressions of unconscious desires. It suggested that dream content might be essentially random, determined more by which neurons happen to fire during REM sleep than by any psychological significance. Hobson later modified his theory, acknowledging that while the initial activation might be random, the synthesis process would necessarily draw on the dreamer's memories, emotions, and concerns, giving dreams a kind of psychological relevance even if they weren't purposefully generated by some deeper aspect of the psyche. Critics of the activation synthesis hypothesis pointed out several problems. First, dreams aren't actually as random as the theory suggests. They often have coherent narratives, recurring themes, and clear connections to the dreamer's waking life. Second, the theory doesn't explain why REM sleep and its associated neural activation exists in the first place. If dreams are just meaningless byproducts, why would evolution have preserved this elaborate mechanism across mammals and birds? Third, the theory struggles to account for dreaming during non-REM sleep, which, while less vivid and frequent, definitely occurs. An alternative perspective emerged from researchers studying memory. Multiple lines of evidence suggested that sleep, particularly REM sleep, plays a crucial role in memory consolidation, the process by which temporary, fragile memories become stable, long-term memories. Studies showed that people deprived of sleep, especially REM sleep, performed worse on memory tests than people allowed to sleep normally. Specific memories appeared to be reactivated during sleep, with patterns of neural activity observed during waking experiences replaying during subsequent sleep. The hippocampus and cortex seem to engage in a dialogue during sleep, with the hippocampus replaying recently formed memories and the cortex gradually incorporating them into its existing knowledge structures. From these observations emerged the memory consolidation theory of dreaming. In this view, dreams are the conscious experience of the memory consolidation process. When the brain reactivates and processes memories during sleep, we experience this as dreaming. The seemingly random jumble of dream content reflects the brain sorting through multiple memories simultaneously, extracting important information, making connections between new and old experiences, and integrating recent events into long-term storage. The emotional content of dreams might reflect the brain's efforts to process emotionally significant experiences, ensuring that emotionally important memories are prioritized for consolidation. This theory has substantial empirical support. Studies have shown that people who learn a task and then sleep show better retention than people who stay awake for an equivalent period. The benefit is even greater when sleep includes REM periods. Specific learned information appears in dreams following training. For instance, people who spent hours navigating a virtual maze reported dreaming about the maze, and those who dreamed about it showed better performance when tested again later. Brain imaging studies have shown that regions activated during learning are reactivated during subsequent REM sleep, and the degree of reactivation correlates with how well the memory is retained. However, the memory consolidation theory also faces challenges. Not all memories appear in dreams, and many dreams include elements that have nothing to do with recent experiences. The theory doesn't easily explain why dreams often feel so bizarre, why they violate logical consistency, or why we forget most of them upon waking. If dreams reflect memory consolidation, why is dream amnesia so common? Some researchers argue that the consolidation process doesn't require consciousness, that the brain could process memories during REM sleep without generating subjective dream experiences, raising the question of why dreams are conscious at all. A third major theory focuses on emotion regulation. Several researchers, notably Matthew Walker and colleagues, have proposed that REM sleep serves to process emotional experiences, particularly negative emotions like fear and anxiety. This emotion regulation theory suggests that dreams provide a kind of overnight therapy, allowing the brain to reprocess emotionally charged experiences in a neurochemical environment that's conducive to emotional depotentiation, the reduction of emotional intensity associated with memories. The key to this process lies in the unique neurochemistry of REM sleep. As mentioned earlier, norepinephrine, a stress-related neurotransmitter associated with the fight or flight response, drops to nearly zero during REM sleep. This creates a state where emotionally charged memories can be reactivated and processed without triggering the full stress response that accompanied the original experience. Over multiple nights, this reprocessing can strip away the emotional charge while preserving the factual content of the memory. You remember what happened, but without the intense emotional reactivity you initially felt. Evidence for emotion regulation comes from multiple sources. Brain imaging studies show that the amygdala, crucial for emotional processing, is highly active during REM sleep, suggesting that emotional content is indeed being processed. People who are deprived of REM sleep show heightened emotional reactivity the next day, reacting more strongly to negative stimuli. Conversely, a good night's sleep, rich in REM, seems to diminish emotional reactivity. Clinical observations of people with post-traumatic stress disorder, who often have disrupted REM sleep and experience recurring nightmares about their trauma, support the idea that normal REM sleep helps process and defuse emotional memories. When REM sleep is disrupted, this emotional processing may fail, leading to persistent emotional distress. The emotion regulation theory helps explain several features of dreams. Dreams are often emotionally intense, featuring fear, anxiety, anger, or joy more prominently than neutral experiences. The bizarre, hallucinatory quality of dreams might actually facilitate emotional processing by creating a safe space where threatening scenarios can be explored without real-world consequences. The theory also explains why we dream about emotionally significant experiences more than mundane ones. The brain is prioritizing emotional content for overnight processing. Yet another perspective comes from evolutionary psychology and the threat simulation theory proposed by Revanchuo. This theory suggests that dreams evolved as a kind of ancient virtual reality training system, allowing our ancestors to practice responding to threats in a safe, simulated environment. Throughout evolutionary history, humans faced numerous survival threats. Predators, hostile neighbors, environmental dangers. Dreams, in this view, function as simulations of threatening scenarios, giving the brain repeated practice at detecting and responding to threats, thereby improving actual threat avoidance during waking hours. Revanchuo points to several lines of evidence. Dream content analysis shows that threatening events are overrepresented in dreams compared to daily waking life. People dream about being chased, attacked, or endangered far more often than they actually experience such events while awake. The amygdala, central to fear processing, is highly active during REM sleep, consistent with the simulation of threatening scenarios. Children, who are most vulnerable and need the most practice at threat avoidance, dream more than adults and report more threatening dream content. Across cultures, similar threatening themes appear in dreams, suggesting these aren't just products of particular social environments but reflect deeper, possibly innate patterns. Critics point out that many dreams don't contain threatening content at all, and even threatening dreams often don't lead to successful threat avoidance within the dream itself. The dreamer might be paralyzed, unable to run from danger, or might wake up before resolving the threat. If dreams were truly rehearsals for threat avoidance, wouldn't they more consistently feature successful escape or defense? Revanchuo's response is that the practice doesn't need to be perfect to be beneficial, and that even failed attempts at threat avoidance in dreams might strengthen relevant neural circuits. A more recent and intriguing theory comes from neuroscientist Eric Hole and colleagues, who proposed the overfitted brain hypothesis. Drawing on concepts from machine learning, Hole suggests that brains, like artificial neural networks, face a fundamental problem. They can become overfitted to their training data. In machine learning, overfitting occurs when a network becomes so specialized for the specific examples it's trained on that it loses the ability to generalize to new situations. A network overfitted to recognize photographs of cats might perform perfectly on its training images but fail when presented with cats in different poses, lighting, or contexts. Hole argues that brains face the same risk. If we only experienced and processed real-world events, our neural networks might become too specialized, too rigid, losing the flexibility needed to handle novel situations. Dreams, in this view, serve as a kind of regularization mechanism, introducing randomness and noise that prevents overfitting. By presenting the brain with bizarre, novel, and unexpected scenarios every night, dreams maintain cognitive flexibility and generalization ability. We don't just learn about the specific dogs we've encountered. We learn about the general category of dog in a way that allows us to recognize novel dogs we've never seen before. This theory has an elegant appeal and makes testable predictions. It suggests that the bizarreness of dreams isn't a bug but a feature, that the incorporation of random, novel elements is precisely what makes dreams useful. It predicts that people with richer, more varied dream lives might show better generalization and creative problem-solving. It explains why dreams often combine familiar elements in unfamiliar ways, creating hybrid creatures, impossible locations, and violations of physical laws. These aren't meaningless distortions, but necessary variations that maintain the brain's flexibility. The overfitted brain hypothesis is still relatively new, and extensive empirical testing is ongoing. One intriguing aspect is that it suggests dreams might be particularly important for species with complex cognitive abilities and long learning periods, exactly the species, like humans and other primates, that show extensive REM sleep. It also connects dreaming to creativity and problem-solving, potentially explaining those famous anecdotes of scientific discoveries and artistic inspirations emerging from dreams. Each of these theories captures something important about dreams, but none seems entirely complete on its own. Memory consolidation clearly occurs during sleep, and dreams likely reflect aspects of this process. Emotional processing appears central to REM sleep's function, and dreams provide the conscious experience of emotional reprocessing. Threat simulation, while perhaps not the only function of dreams, might explain some recurring themes and the prominence of the amygdala in dreaming. The overfitted brain hypothesis offers a compelling framework for understanding dream bizarreness and its potential cognitive benefits. Perhaps the most accurate view is that dreams serve multiple functions simultaneously. The sleeping brain is accomplishing many things: consolidating memories, processing emotions, maintaining neural flexibility, and possibly rehearsing responses to challenges. Dreams are our conscious window into these multiple processes, a kind of mental screen where the brain's various nighttime activities become subjectively experienceable. Different dreams might emphasize different functions at different times, depending on recent experiences, emotional state, and current concerns. What remains clear is that dreams are not meaningless noise, not random mental static to be dismissed or ignored. They emerge from crucial brain processes that support learning, emotional health, and cognitive flexibility. Understanding dreams requires understanding sleep itself, understanding memory and emotion, understanding consciousness and how the brain generates subjective experience from objective neural activity. The mystery hasn't been fully solved, but the revolution begun in Aserinsky's laboratory has provided tools and frameworks that continue to deepen our understanding of these nightly mental adventures. The chemistry of dreams. The brain is fundamentally a chemical organ. Its 100 billion neurons communicating not only through electrical impulses, but through an intricate symphony of chemical signals. To truly understand dreaming, we must delve into the neurochemical transformations that occur as we transition from wakefulness through various sleep stages and into the dream-rich REM periods. These chemical shifts are not mere side effects but active orchestrators of the changes in consciousness, perception, and cognition that characterize sleep and dreams. The story of dream chemistry is the story of neurotransmitters rising and falling, hormones surging and receding, creating the precise biochemical conditions that make dreams possible. Let's begin with acetylcholine, perhaps the most important neurotransmitter for understanding REM sleep and dreaming. Acetylcholine is ubiquitous throughout the brain and body, involved in everything from muscle control to memory formation to maintaining attention. In the brain, cholinergic neurons, those that release acetylcholine, are particularly concentrated in the basal forebrain and in the brainstem regions we discussed earlier, the pedunculopontine tegmentum and laterodorsal tegmentum. These brainstem cholinergic neurons are the conductors of the REM sleep orchestra, their activity initiating the cascade of changes that define this sleep stage. During waking hours, acetylcholine levels in the cortex are elevated, supporting the alert, attentive state necessary for processing sensory information and responding to environmental demands. As we fall asleep and descend into non-REM sleep, particularly the deep, slow-wave sleep of early night, acetylcholine levels drop substantially. The cortex becomes relatively quiescent, disconnected from both external sensory input and internal activation. But then, as the first REM period approaches roughly 90 minutes into sleep, something remarkable happens. Acetylcholine levels surge back to waking levels, sometimes even exceeding them, flooding the cortex and hippocampus with cholinergic signals. This surge of acetylcholine during REM sleep has multiple crucial effects. First, it activates the thalamus, which then sends signals to the cortex, creating the activated, wake-like EEG patterns characteristic of REM sleep. Second, it triggers the pontogeniculo-occipital waves, or PGO waves, those electrical bursts that propagate from the pons through the lateral geniculate nucleus of the thalamus to the occipital visual cortex. These PGO waves are thought to generate much of the visual imagery of dreams, creating the internally generated perception of scenes, objects, and faces that populate our dream worlds. Third, acetylcholine in the hippocampus facilitates memory processes, potentially contributing to the incorporation of recent experiences into dream content and supporting the memory consolidation functions of REM sleep. The high acetylcholine environment of REM sleep might also explain certain cognitive features of dreams. Acetylcholine enhances the processing of internal mental representations while reducing the influence of external sensory input, creating a state where imagination dominates over perception. This internal focus, combined with activation of sensory cortices, generates the hallucinatory quality of dreams, where internally generated images seem as real as waking perception. Interestingly, drugs that enhance acetylcholine activity, such as galantamine, can increase dream vividness and even promote lucid dreaming, further supporting acetylcholine's central role in dream generation. Now let's turn to the monoamines: serotonin, norepinephrine, and dopamine. These neurotransmitters show a dramatically different pattern across the sleep-wake cycle compared to acetylcholine, and their absence during REM sleep is just as important as acetylcholine's presence. During waking hours, neurons in specific brainstem nuclei, the raphe nuclei for serotonin, the locus coeruleus for norepinephrine, and the ventral tegmental area for dopamine fire steadily, releasing their respective neurotransmitters throughout the brain. These monoaminergic systems support wakefulness, attention, and emotional regulation. They help maintain the boundaries between imagination and reality, between internal mental events and external perceptions. As we transition into non-REM sleep, activity in these monoaminergic neurons decreases. During slow-wave sleep, their firing slows to about half the waking rate. But during REM sleep, something unprecedented occurs. They fall almost completely silent. Serotonin and norepinephrine levels drop to near zero. The brain enters a unique neurochemical state that exists nowhere else across the 24-hour cycle, a state characterized by high acetylcholine and virtually absent monoamines. This absence of norepinephrine during REM sleep appears particularly important for several aspects of dreaming. Norepinephrine is closely associated with stress responses, memory consolidation, and the encoding of emotionally arousing experiences. Its near-complete absence during REM sleep may create the ideal conditions for emotional memory reprocessing without the stress response that originally accompanied the memories. Matthew Walker and colleagues have proposed that this norepinephrine-free state allows the brain to revisit emotional memories, reactivating the factual content while diminishing the emotional charge, effectively providing overnight therapy for emotional experiences. The absence of norepinephrine might also contribute to dream amnesia, the frustrating phenomenon where vivid dreams are rapidly forgotten upon waking. Norepinephrine plays a crucial role in memory encoding and consolidation. When it's absent, as during REM sleep, memories formed during that state may be particularly fragile and prone to rapid forgetting. This could be adaptive, preventing our minds from becoming cluttered with memories of experiences that never actually occurred, keeping the boundary between dreams and reality relatively clear despite the fact that dreams feel completely real while we're experiencing them. Dopamine's role in REM sleep and dreaming is more complex and less well understood than that of acetylcholine, serotonin, or norepinephrine. Unlike serotonin and norepinephrine, dopamine doesn't completely disappear during REM sleep. Dopaminergic activity, particularly in limbic regions associated with reward and motivation, may actually increase during REM sleep. This has led some researchers to propose that dopamine might contribute to the motivational and emotional aspects of dreams. The sense that dream events matter, that they're worth pursuing or avoiding. Interestingly, the neurochemistry of REM sleep, particularly the combination of high cholinergic and low monoaminergic activity coupled with elevated dopamine in certain regions, bears a striking resemblance to the neurochemistry associated with psychosis and hallucinations. People experiencing psychotic episodes often show altered dopamine function and may have disturbed sleep and vivid, often frightening hallucinations. Some researchers have suggested that dreams represent a kind of temporary, healthy psychosis, a state where we experience hallucinations, delusions, and cognitive disorganization but within the safe confines of sleep, from which we reliably wake each morning with our grasp on reality restored. This comparison, while somewhat speculative, highlights how dramatically different the dreaming brain is from the waking brain, chemically and experientially. Beyond neurotransmitters, hormones also show important changes across sleep stages, and these hormonal shifts interact with neurotransmitter systems to shape the sleeping and dreaming brain. Cortisol, the body's primary stress hormone, follows a strong circadian rhythm, with levels lowest during early night and rising steadily through the later sleep period, peaking just before and after normal wake time. This rising cortisol tide coincides with the longer, more intense REM periods of late night and early morning. Some researchers have proposed that cortisol might modulate emotional memory processing during REM sleep, with the increasing cortisol levels of late night potentially contributing to the processing of emotionally negative memories. Melatonin, often called the hormone of darkness, shows the opposite pattern to cortisol. Melatonin is produced by the pineal gland in response to darkness, rising sharply in the evening, remaining elevated throughout the night, and falling again as dawn approaches. Melatonin doesn't just promote sleepiness. It appears to influence dream content as well, potentially increasing dream vividness and frequency. People taking melatonin supplements often report more vivid and memorable dreams, though the mechanism remains unclear. Melatonin might enhance REM sleep directly, prolong REM periods, or influence neurotransmitter systems involved in dream generation. Growth hormone shows yet another pattern, surging during deep slow-wave sleep in the first half of the night. While less directly related to dreaming than cortisol or melatonin, growth hormones released during sleep underscores that sleep is a time of active physiological restoration and regulation, not passive downtime. The hormonal environment of sleep supports not just neural processes like memory consolidation and emotional regulation, but also physical restoration, tissue repair, and metabolic regulation. The neurochemical state of the dreaming brain also involves changes in neuropeptides, small protein-like molecules that neurons use for signaling. Orexin, also called hypocretin, which we discussed in relation to narcolepsy, is one example. Orexin neurons are active during waking and quiet during both REM and non-REM sleep. Their silence is essential for sleep to occur, and their activity is crucial for maintaining stable wakefulness. The loss of orexin neurons in narcolepsy leads not just to excessive daytime sleepiness, but also to phenomena where REM sleep intrudes into waking, such as cataplexy, sudden muscle weakness during waking that resembles REM atonia, and hypnagogic hallucinations, vivid dreamlike experiences occurring while falling asleep or waking up. These symptoms reveal how tightly orexin regulates the boundaries between sleeping and waking, between dreaming and waking consciousness. GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter, also plays crucial roles in sleep regulation. GABAergic neurons in the ventrolateral preoptic area of the hypothalamus actively inhibit arousal systems during sleep, essentially putting the brakes on the neural systems that promote wakefulness. Without this active inhibition, sleep wouldn't occur reliably. People with insomnia often show reduced GABAergic function, and many sleep medications work by enhancing GABA activity, increasing inhibition of arousal systems to promote sleep onset and maintenance. This complex neurochemical ballet, with different neurotransmitters and hormones rising and falling across sleep stages, creates the precise conditions necessary for the various functions of sleep. The high acetylcholine, low monoamine state of REM sleep appears specifically tuned for emotional memory reprocessing, memory consolidation, and the generation of dreams themselves. The slow-wave sleep that dominates early night, with its low acetylcholine and reduced monoamines, creates optimal conditions for declarative memory consolidation, the strengthening of factual and episodic memories. The transitions between sleep stages, mediated by shifts in neurotransmitter and hormone levels, ensure that we cycle through these different states multiple times each night, each cycle contributing to the restorative functions of sleep. Understanding dream chemistry also has practical implications. Many medications influence neurotransmitter systems and therefore potentially affect dreaming. Antidepressants that increase serotonin levels often suppress REM sleep and reduce dream recall, sometimes eliminating nightmares but potentially interfering with important emotional processing. Beta blockers used to treat high blood pressure can increase nightmares, possibly by affecting norepinephrine systems. Medications for Parkinson's disease that boost dopamine can intensify dreams and increase the risk of disturbing nightmares. Even common substances like alcohol and marijuana alter sleep architecture and dream experiences, typically suppressing REM sleep in the short term and leading to REM rebound, more intense dreams and nightmares, when their use is discontinued. The neurochemistry of dreams reminds us that consciousness itself, whether waking or sleeping, emerges from chemical processes in the brain. The subjective richness of dream experiences, the visual beauty or terror, the emotional intensity, the narrative complexity, all arise from patterns of neurotransmitter release, receptor activation, and neural firing regulated by these chemical signals. Understanding these chemical foundations doesn't diminish dreams but enriches our appreciation of how the brain creates the full spectrum of conscious experience, from the clearest waking perception to the strangest dream. Each state of consciousness has its own chemical signature, its own neurobiological underpinnings, and dreams occupy a unique place in the spectrum, a state as chemically distinctive as it is experientially remarkable. Dreams and memory, the night shift. Every night, as you surrender to sleep, your brain begins one of its most crucial tasks: organizing, consolidating, and integrating the countless experiences and pieces of information you accumulated during the waking day. This is memory consolidation, the process by which fleeting impressions are transformed into lasting knowledge, and sleep plays an indispensable role. Dreams, in this context, aren't just entertainment for the sleeping mind but may be the conscious reflection of memory's night shift, the experience of watching, or rather living through, the brain's efforts to make sense of what you've learned and experienced. The scientific evidence linking sleep to memory consolidation is overwhelming and comes from multiple experimental approaches. The most straightforward studies compare memory performance in people who sleep after learning to those who stay awake for an equivalent period. Across hundreds of studies examining different types of memory, the pattern is clear. Sleep helps. People who sleep after learning a list of word pairs, a sequence of finger movements, a spatial layout, or any of countless other tasks show better retention when tested later compared to people who stay awake. This benefit isn't just about being rested versus fatigued. Even when researchers control for fatigue, match circadian timing, and account for other factors, sleep itself enhances memory consolidation. More sophisticated studies have investigated which stages of sleep contribute most to different types of memory. The picture that emerges is complex but revealing. Declarative memories, the conscious memories for facts and events, things you can talk about and describe, appear to benefit particularly from slow-wave sleep, that deep sleep rich in high-amplitude delta waves that dominates the first half of the night. Procedural memories, the unconscious memories for skills and habits like riding a bicycle or typing, seem to depend more on REM sleep and stage 2 sleep. Emotional memories, those tagged with strong feelings, show enhanced consolidation during REM sleep, when the amygdala is highly active and emotional content can be reprocessed. But how exactly does sleep enhance memory? What's happening in the sleeping brain that makes memories more stable, more accessible, more integrated? To answer this, we need to return to the hippocampus, that seahorse-shaped structure so crucial for forming new memories. During waking experience, the hippocampus rapidly encodes new information, creating sparse, pattern-separated neural representations that keep similar experiences distinct from one another. These hippocampal representations are initially fragile. They can be disrupted by interference from subsequent experiences or by simple decay over time. For memories to last, they need to be transferred from the hippocampus to the neocortex, distributed across wide cortical networks where they can be stored more permanently and integrated with existing knowledge. This transfer process, called systems consolidation, occurs primarily during sleep, particularly during slow-wave sleep. During waking hours, information flows from sensory cortices through the hippocampus and back to cortex, with the hippocampus binding together disparate cortical elements into coherent memories. But during sleep, this flow reverses. The hippocampus spontaneously reactivates recently formed memories, replaying the patterns of neural activity that occurred during encoding. This replay is not random but selective, preferentially reactivating memories that were emotionally significant or that the brain has been explicitly cued to remember. Researchers have directly observed this memory replay in animals. By recording from large populations of neurons in the hippocampus while rats explored a maze, scientists identified specific sequential patterns of neural firing associated with specific paths through the maze. Then, during subsequent sleep, they observed these exact same sequences replaying, often in rapid, compressed form. A path that took the rat 20 seconds to traverse might replay in just 200 milliseconds. These compressed replays occur during sharp wave ripples, brief high-frequency bursts of neural activity in the hippocampus that punctuate slow-wave sleep. Each ripple might contain a complete replay of a learned sequence, and thousands of ripples occur over a night's sleep, providing countless opportunities for memory consolidation. As the hippocampus replays memories, it sends these reactivated patterns to the cortex, particularly to the medial prefrontal cortex, an area thought to coordinate the distribution of memories across neocortical networks. The cortex, receiving these replay signals from the hippocampus, gradually builds its own representations of the memories, integrating new information with previously stored knowledge, extracting general principles and patterns from specific experiences, and developing the stable, distributed neural networks that support long-term memory storage. Through repeated reactivation across multiple nights, memories that initially depended on the hippocampus become gradually independent of it, supported instead by corticocortical connections. This is why damage to the hippocampus typically affects recent memories more than remote ones. Recent memories haven't yet completed the consolidation process and still depend on hippocampal involvement, while older memories have been distributed to cortex and can survive hippocampal damage. REM sleep contributes to memory consolidation through different mechanisms. During REM, the hippocampus remains active, and theta oscillations, rhythmic patterns of neural activity in the 4 to 8 Hz range, dominate hippocampal recordings. These theta oscillations appear to support the binding of different elements of memories and may be particularly important for emotional memory consolidation. The high acetylcholine levels and activated state of the cortex during REM create conditions favorable for synaptic plasticity, the strengthening or weakening of connections between neurons that underlies learning and memory at the cellular level. Evidence from studies of lucid dreaming, those rare occasions when dreamers become aware they're dreaming while remaining asleep, provides a unique window into the relationship between dreams and memory processing. In lucid dreaming studies, researchers can communicate with sleeping subjects using predefined eye movement signals. By asking lucid dreamers to rehearse specific motor sequences during their dreams, researchers have shown that such mental practice during REM sleep produces neural activation patterns similar to actual physical practice and can improve subsequent waking performance. The brain, it seems, doesn't sharply distinguish between vividly imagined experiences during dreams and real experiences, at least in terms of how it processes and consolidates skills. But what about the subjective experience of dreaming itself? How does this relate to memory consolidation? Several lines of evidence suggest that dream content directly reflects the memory consolidation process occurring during sleep. First, recent experiences, particularly novel or emotionally significant ones, frequently appear in dreams. In one study, people who spent hours playing the video game Tetris, watching colored blocks fall and rotate, frequently reported dreaming about falling blocks, seeing Tetris shapes, or feeling the cognitive processes of rotating blocks. Interestingly, even amnesic patients with severe hippocampal damage who couldn't consciously remember playing Tetris still reported Tetris-related dream imagery, suggesting that some aspects of memory processing during sleep can occur even without normal hippocampal function. Second, the timing of when specific memories appear in dreams reveals something about the consolidation process. Researchers have identified two windows when recent experiences are most likely to appear in dreams. The first or second night after an experience reflecting immediate consolidation, and about a week later, when the memory is being integrated more fully into long-term storage. This pattern suggests dreams provide a window into both initial encoding and later consolidation stages. Third, the relationship between dream content and subsequent memory performance supports the idea that dreams reflect active consolidation. In studies where people learn a spatial task and then nap, those who report dreaming about the task show greater improvement after the nap than those who either don't dream or dream about unrelated content. This doesn't necessarily mean that dreaming causes better memory. The causality could run the other way, with more successful consolidation producing more task-related dreams. But it suggests that what we dream about isn't arbitrary but reflects what the brain is actively working on during sleep. The incorporation of memories into dreams isn't literal or straightforward. Specific details might be altered, contexts changed, elements from different experiences combined. This seeming distortion might actually be functional. Memory consolidation isn't just about preserving exact copies of experiences, but about extracting meaningful patterns, making connections between related information, and integrating new learning into existing knowledge structures. The bizarre combinations and transformations characteristic of dreams might reflect this synthetic, integrative aspect of memory consolidation. The brain is figuring out what the new information means, how it relates to what's already known, what general principles can be extracted. This connects to the overfitted brain hypothesis we discussed earlier. By presenting the brain with novel combinations and variations on learned material, dreams might help prevent the overspecificity that could result from just repeatedly replaying exact experiences. The Tetris players didn't just dream about the exact sequences of blocks they'd encountered. They dreamed about variations, novel combinations, impossible scenarios involving Tetris elements. This variability might help the brain learn the general principles of the task, the category Tetris, rather than just memorizing specific instances. Sleep and dreams also appear crucial for insight and problem-solving, particularly problems that benefit from finding non-obvious connections or restructuring knowledge in novel ways. The famous story of Auguste Kekulé discovering the ring structure of benzene after dreaming of a snake biting its tail is perhaps apocryphal, but controlled laboratory studies have confirmed that sleep can enhance insight. In one study, people working on a number reduction task that had a hidden rule that, once discovered, made the task much easier were 3 times more likely to discover this hidden rule after sleeping than after an equivalent waking interval. The sleeping brain, freed from the constraints of logical, sequential waking thought, might explore solution spaces more creatively, making connections that elude the waking mind. The role of dreams in emotional memory consolidation deserves special emphasis. Emotional experiences are prioritized for consolidation during sleep, tagged by stress hormones like cortisol and adrenaline as particularly important to remember. But as we discussed in the chemistry section, REM sleep provides a unique environment for processing emotional memories, reactivating them in the absence of norepinephrine, allowing the factual content to be strengthened while the emotional reactivity is diminished. Dreams often contain emotional content, sometimes intensely so, and this emotional content may reflect the brain's active processing of emotional experiences. Studies examining post-traumatic stress disorder provide tragic evidence for the importance of this emotional processing. PTSD patients often have fragmented, disrupted sleep, particularly REM sleep, and they experience recurring nightmares about their traumatic experiences. These nightmares differ from normal dreams. Rather than integrating and processing the trauma, moving toward resolution, PTSD nightmares often replay the trauma repeatedly without resolution. The normal REM sleep processes that should help integrate and defuse traumatic memories appear to be disrupted in PTSD, leading to persistent emotional reactivity and intrusive memories. Treatments that improve sleep quality and reduce nightmare frequency often also reduce PTSD symptoms, supporting the idea that proper sleep-dependent emotional processing is crucial for emotional health. For healthy individuals, the picture is more positive. Each night's dreams appear to contribute to emotional equilibrium, processing the emotional ups and downs of daily life, helping us wake each morning with a somewhat fresh emotional slate, ready to handle new challenges without being overwhelmed by yesterday's emotions. The phrase, "sleep on it," when facing an emotional decision isn't just folk wisdom, but reflects an intuitive understanding of sleep's role in emotional processing. After sleep, especially sleep-rich in REM and dreams, emotional problems often seem more manageable, emotional reactions less intense, and solutions more apparent. The relationship between sleep, dreams, and memory isn't simple or one-directional. Different types of memory benefit from different sleep stages, and dreams reflect the complex, multi-layered processing occurring throughout the night. But the evidence is clear. Dreams aren't just random mental noise, but are deeply connected to one of the brain's most crucial functions, the transformation of fleeting experience into lasting knowledge, integrated with existing understanding and stripped of inappropriate emotional reactivity. Every night, as you dream, your brain is working hard, making sense of your experiences, preparing you for tomorrow, and building the continuity of self that makes you who you are. The emotional workshop. Close your eyes and recall your most vivid dream from recent memory. Chances are that dream contained powerful emotions. Perhaps you felt fear, running from some nameless threat through impossible landscapes. Perhaps you experienced joy, reuniting with a loved one or achieving something magnificent. Or maybe anxiety pervaded the dream, tasks left undone, deadlines approaching, preparations failing. Dreams are soaked in emotion, far more so than waking thought, and this emotional intensity isn't accidental. It reflects one of the most important, least appreciated functions of sleep: emotional regulation, the overnight recalibration of our emotional responses, and the integration of emotional experiences into our psychological landscape. The emotional intensity of dreams is striking when you analyze dream content systematically. Studies where people are awakened during REM sleep and asked to describe their dreams reveal that more than 70% of dreams contain clearly identifiable emotions, with negative emotions like fear, anxiety, and anger appearing somewhat more frequently than positive ones. This negativity bias isn't universal. Dreams can certainly be pleasant, joyful, even ecstatic. But the prevalence of negative emotions in dreams appears to reflect a kind of overnight emotional triage, with the brain prioritizing potentially threatening or problematic experiences for processing. The neurobiological basis for this emotional processing centers on the amygdala and its interactions with the hippocampus and prefrontal cortex during sleep. The amygdala, that almond-shaped structure buried deep in the temporal lobe, serves as the brain's emotional alarm system, particularly for threat-related emotions like fear and anxiety. During waking hours, the amygdala processes incoming sensory information for emotional significance, rapidly identifying potential threats before conscious awareness even kicks in. This is why you might jump at a shadow or a sudden sound before consciously recognizing what it is. The amygdala has reacted first, triggering defensive responses that can save your life if the threat is real. During REM sleep, the amygdala doesn't rest. In fact, brain imaging studies show that the amygdala is even more active during REM sleep than during waking, and it's connected differently to other brain regions. Normally, during waking hours, the medial prefrontal cortex exerts some regulatory control over the amygdala, dampening responses that might be excessive or inappropriate to the actual level of threat. But during REM sleep, the prefrontal cortex is relatively deactivated, and this top-down regulation is diminished. The amygdala is essentially running free, processing emotional content without the usual cognitive oversight. This might sound like a problem, an overactive emotional brain without rational oversight. But researchers have proposed that this is precisely the state needed for emotional memory processing. The unique neurochemical environment of REM sleep, particularly the near-complete absence of norepinephrine, creates conditions where emotional memories can be reactivated without triggering the full stress response that accompanied the original experience. The amygdala can replay emotionally charged memories, strengthening the memory trace, the what happened part, while the absence of stress neurochemistry prevents the re-encoding of intense emotional arousal. Matthew Walker and Els van der Helm at UC Berkeley have championed what they call the sleep to forget and sleep to remember model, often abbreviated as SFSR. According to this model, REM sleep serves a kind of overnight therapy function, allowing us to remember the facts of emotional experiences while forgetting, or at least diminishing, the emotional pain or reactivity associated with them. Over successive nights of sleep, particularly REM sleep, emotionally charged memories undergo a transformation. The content remains accessible, you can remember what happened. But the emotional sting fades, the memory becomes less likely to trigger strong emotional reactions, and you gain some psychological distance from the experience. Evidence for this model comes from multiple sources. In laboratory studies, people shown emotionally disturbing images and then tested the next day show reduced amygdala reactivity when viewing the same images again, but only if they slept in the interim. People who stayed awake showed no such reduction, and people who slept but whose REM sleep was experimentally disrupted showed less benefit than those with normal REM sleep. The reduction in amygdala reactivity correlated with REM sleep duration and quality, suggesting a dose-response relationship. More and better REM sleep produced more emotional dampening. Brain imaging during REM sleep has revealed the mechanism more directly. During REM sleep periods, researchers observed increased connectivity between the amygdala and the medial prefrontal cortex, despite the prefrontal cortex's overall deactivation. This connectivity appeared to reflect the prefrontal cortex gradually building representations of emotional memories, taking over some of the amygdala's role, allowing the cortical networks to provide context and regulate emotional responses rather than having the amygdala trigger reflexive emotional reactions. Over time and across multiple nights, this process transfers emotional memories from the amygdala's immediate, reactive processing to the cortex's more nuanced, context-sensitive processing. The emotional processing function of REM sleep appears particularly crucial for mental health. Disrupted sleep, especially disrupted REM sleep, is associated with mood disorders, anxiety disorders, and emotional dysregulation. Depression almost always involves sleep disturbances, typically including early morning awakening and altered REM sleep patterns. Anxiety disorders similarly involve sleep problems, often with difficulty falling asleep and frequent nighttime awakenings. Whether these sleep disturbances are cause or consequence of the mood problems is unclear. The relationship is likely bidirectional. But improving sleep often improves mood, and interventions that normalize REM sleep can reduce emotional symptoms. The tragic case of post-traumatic stress disorder provides perhaps the clearest evidence for REM sleep's role in emotional processing. PTSD develops when normal emotional memory processing fails, leaving the individual with vivid, intrusive memories of traumatic events that retain their full emotional intensity despite the passage of time. PTSD patients typically have severely disrupted sleep, with frequent awakenings, difficulty falling back asleep, and a high prevalence of nightmares. These nightmares differ from ordinary dreams in critical ways. Rather than transforming and integrating traumatic content, PTSD nightmares often replay the trauma with little variation, and these replays don't lead to resolution or reduction in emotional intensity. Instead, they seem to re-traumatize the person each night. Research has shown that the normal REM sleep neurochemistry is disrupted in PTSD. Norepinephrine levels, which should drop to near zero during REM sleep, remain elevated in PTSD patients. This persistence of stress neurochemistry during REM sleep may prevent the normal emotional depotentiation process. The brain is trying to process the traumatic memory. That's why it keeps appearing in dreams. But the neurochemical conditions necessary for successful processing aren't present. The memory is reactivated but not transformed, leading to a kind of emotional stuck state. Treatments for PTSD often target sleep and nightmares specifically. Prazosin, a medication that blocks norepinephrine receptors, has shown promise in reducing PTSD nightmares and improving sleep quality. By artificially creating the low norepinephrine environment that should occur naturally during REM sleep, prazosin may allow normal emotional processing to resume. Imagery rehearsal therapy, a psychological treatment where patients rehearse altered versions of their nightmares during waking hours, imagining different, less threatening outcomes, also shows effectiveness. By providing an alternative neural pathway for processing the traumatic content, this therapy may help the brain integrate the trauma rather than repeatedly reliving it. But emotional processing during sleep isn't only about negative emotions or traumatic experiences. Dreams also appear to help us process positive emotions, rewarding experiences, and emotionally meaningful interactions. The consolidation of positive memories, particularly those involving social connection, reward, and achievement, may help maintain motivation, reinforce adaptive behaviors, and support psychological well-being. Dreams about positive experiences might feel less urgent or memorable than nightmares, but they likely contribute importantly to our overall emotional balance. There's also evidence that dreams may help with specific emotional challenges or decisions. Anecdotal reports of people gaining clarity about difficult decisions after dreaming about them are common. While controlled studies are difficult to conduct, some research suggests that sleep, particularly REM sleep, can facilitate emotional problem-solving. By allowing the brain to explore emotional scenarios, consider different outcomes, and integrate emotional information with factual knowledge, dreams might help us navigate complex emotional landscapes that seem overwhelming when approached with waking logic alone. The content of dreams reflects this emotional processing work. Dreams often take elements from recent experiences and recombine them with older memories, personal concerns, and emotional themes. A stressful interaction at work might appear in a dream, but transformed, perhaps combined with elements from childhood or rendered in a completely different setting. These transformations aren't meaningless distortions but may reflect the brain's efforts to extract the emotional significance of the experience, to connect it with similar past experiences, and to develop more generalized emotional responses that can guide future behavior. This connects to what psychotherapists have long observed. Paying attention to dreams can provide insight into emotional concerns, unresolved conflicts, and psychological dynamics that may be less apparent during waking consciousness. While the Freudian notion that dreams disguise forbidden wishes through symbolic distortion has largely fallen out of favor, the broader idea that dreams reflect emotional processing and can reveal concerns that deserve attention remains influential in many psychotherapy approaches. Modern dream work in therapy doesn't typically involve decoding supposed universal symbols, but rather asks what the dream's emotional content and themes reveal about the dreamer's current concerns and psychological state. There's also interesting research on the relationship between daytime mood and subsequent dream content, and between dream content and next-day mood. People experiencing stress or negative emotions during the day tend to have more negative dream content, suggesting the brain is working on these emotions overnight. Conversely, dream content can predict next-day mood, with more negative dreams associated with more negative waking mood, while positive dreams can enhance waking mood and well-being. This bidirectional relationship supports the idea that dreams are part of an ongoing process of emotional regulation that spans both sleep and waking life. The question of why emotional processing should occur during sleep rather than, or in addition to, during waking hours is intriguing. One possibility is that sleep provides a safe space for emotional processing, disconnected from the demands and potential threats of the waking environment. During waking hours, we often suppress emotional processing in favor of goal-directed behavior, dealing with immediate demands. We don't have time or safety to fully process difficult emotions when we're navigating social situations, meeting work demands, or handling practical tasks. Sleep provides dedicated processing time when the brain can focus on emotional integration without the need to maintain vigilant awareness of the environment or respond to immediate challenges. Another factor is the unique neurochemical environment of REM sleep. The absence of norepinephrine creates conditions that simply don't exist during waking hours. This allows for a kind of emotional processing reactivation without re-traumatization that wouldn't be possible when stress neurochemistry is present. The deactivation of the prefrontal cortex during REM sleep might also be relevant, reducing cognitive censorship and allowing more direct emotional processing without the overlay of rational evaluation or social appropriateness that might constrain waking emotional processing. Whatever the full explanation, the evidence is clear. Dreams are deeply involved in emotional life, not just reflecting our emotions but actively helping to process them, integrate them, and regulate them. Each night's dreams contribute to our emotional balance, helping us wake with a somewhat reset emotional slate, ready to face new challenges without being overwhelmed by yesterday's emotional residue. Understanding this emotional workshop function of dreams helps explain why sleep deprivation is so devastating to mood and emotional regulation, why mental health problems almost always involve sleep disturbances, and why paying attention to dreams might offer valuable insights into our emotional lives. The sleeping brain is hard at work on one of life's most crucial tasks: making sense of our emotional experiences and maintaining the psychological equilibrium necessary for mental health and well-being. When dreams go wrong, for most people, most of the time, dreams are a benign and even beneficial part of sleep. They may be strange, occasionally unsettling, but they don't typically disrupt sleep or cause lasting distress. But for some people, some of the time, dreams and sleep-related experiences can go seriously wrong, leading to nightmares, sleep terrors, paralysis, or dangerous behaviors during sleep. Understanding these disorders provides unique insights into the mechanisms of normal dreaming, while also highlighting the potential consequences when those mechanisms malfunction. Nightmares are probably the most familiar form of disturbing dream experiences. Everyone has occasional nightmares, frightening dreams that wake us with hearts racing and lingering anxiety. Occasional nightmares are completely normal and likely represent the brain's processing of threatening or anxiety-provoking content. But for some people, nightmares occur frequently, perhaps several times per week, causing significant distress and sleep disruption. When nightmares are this frequent and disruptive, the condition is called nightmare disorder, recognized as a legitimate sleep disorder requiring treatment. Nightmare disorder affects about 4% of adults, though prevalence is higher in children and in people with certain psychiatric conditions, particularly post-traumatic stress disorder. Nightmare disorder dreams are characterized by elaborate, story-like narratives featuring threats to survival, security, or physical integrity. The dreamer typically feels intense fear, horror, or anxiety, and these emotions persist even after waking, making it difficult to return to sleep. Unlike night terrors, which we'll discuss shortly, people with nightmare disorder remember their frightening dreams in vivid detail, and this memory of the threat contributes to their distress. What causes nightmare disorder? Several factors appear to contribute. Some people may have a biological vulnerability, perhaps related to how their amygdala responds to potential threats or how their REM sleep is regulated. Stress, anxiety, and depression increase nightmare frequency, as do certain medications, particularly some antidepressants, blood pressure medications, and drugs affecting dopamine. Trauma, whether recent or remote, can trigger recurring nightmares, sometimes featuring literal replays of traumatic events, but often incorporating traumatic themes in more symbolic or transformed ways. Treatment for nightmare disorder often involves imagery rehearsal therapy, where patients practice reimagining their nightmares during waking hours, changing the ending or some element to make the dream less threatening. This therapy appears to work by creating new neural pathways for processing the nightmare content, giving the brain an alternative to the recurring frightening narrative. Other treatments include prazosin for nightmares associated with PTSD, various psychotherapeutic approaches to address underlying anxiety or trauma, and sleep hygiene interventions to improve overall sleep quality, which can reduce nightmare frequency. Lucid dreaming, those rare experiences where dreamers become aware they're dreaming while remaining asleep, has been explored as both a potential cause and a potential treatment for nightmares. Some research suggests that training people to achieve lucid dreams might allow them to consciously alter frightening dream content, turning nightmares into less threatening or even positive experiences. However, the evidence is mixed, and lucid dreaming itself can be associated with sleep disruption and other unusual sleep experiences. Lucid dreaming is fascinating from both scientific and experiential perspectives. During lucid dreams, dreamers report waking-like awareness and sometimes volitional control while remaining physiologically asleep, particularly in REM sleep. Brain imaging and EEG studies of lucid dreaming show a unique pattern of brain activation, with regions of the prefrontal cortex that are typically deactivated during normal REM sleep showing increased activity during lucid dreams. This suggests that lucid dreaming represents a hybrid state, combining features of REM sleep with aspects of waking consciousness, particularly the metacognitive awareness that we're experiencing our own mental states. Lucid dreaming occurs spontaneously in about half the population at least occasionally, and about 20% of people report lucid dreaming at least once per month. Certain techniques can increase lucid dreaming frequency, including reality testing during waking hours, asking yourself repeatedly whether you're dreaming to create a habit that might carry over into dreams, setting intentions before sleep to recognize when you're dreaming, and using the wake back to bed technique, where you wake after several hours of sleep and then return to sleep while holding the intention to become lucid. While lucid dreaming is generally considered a benign phenomenon, it has been associated with some potentially problematic sleep experiences. People who frequently lucid dream appear to have higher rates of sleep paralysis, an experience where consciousness returns before the muscle paralysis of REM sleep has lifted, leaving the person temporarily unable to move despite being awake. Sleep paralysis is often frightening, sometimes accompanied by hallucinations of threatening presences in the room, difficulty breathing, or feelings of chest pressure. These hallucinations likely result from the hybrid state of consciousness, part awake and part still dreaming, with the amygdala active and generating fear in response to the paralysis, but the perceptual systems creating quasi-dream experiences that feel completely real. Sleep paralysis is surprisingly common, experienced by about 8% of the population at some point in their lives. It typically lasts only seconds to a couple of minutes before normal movement returns, and while frightening, it's not dangerous. Understanding what's happening, recognizing that this is a known phenomenon with a physiological explanation, can reduce the fear associated with sleep paralysis. Some sleep paralysis sufferers report that focusing on trying to move just a single finger or toe can help break the paralysis more quickly. Moving from disturbing conscious experiences during sleep to problematic behaviors during sleep, we encounter the parasomnias, a category of sleep disorders characterized by abnormal movements, behaviors, emotions, perceptions, or autonomic nervous system activity during sleep or sleep-wake transitions. Parasomnias are broadly divided into those occurring during non-REM sleep and those occurring during REM sleep. Non-REM parasomnias, also called disorders of arousal, include sleepwalking, night terrors, and confusional arousals. These typically occur during deep slow-wave sleep, when cortical activity is reduced and conscious awareness is minimal. In sleepwalking, individuals engage in complex behaviors while remaining largely asleep, potentially walking around, eating, or even leaving the house, all while having little or no awareness of their actions and typically no memory of them the next morning. Night terrors involve sudden arousal from slow-wave sleep with intense fear, often accompanied by screaming, thrashing, and signs of autonomic arousal like rapid heartbeat and sweating. Unlike nightmares, which people remember, night terrors are typically not remembered, and the person often has no dream narrative to explain their fear. Confusional arousals involve partial awakening with disorientation, slow thinking, and sometimes inappropriate or bizarre behavior. Non-REM parasomnias are more common in children than adults, affecting perhaps 15 to 40% of children at some point, though most outgrow them. In adults, they're less common but can be triggered by sleep deprivation, stress, alcohol, or certain medications. Treatment typically focuses on safety measures, ensuring the environment is safe to prevent injury during episodes, improving sleep hygiene to reduce sleep deprivation that might trigger events, and managing stress. Medications are rarely necessary but can be used in severe cases. REM sleep parasomnias are different in character and mechanism. The most significant is REM sleep behavior disorder, or RBD, where the normal muscle paralysis of REM sleep fails, allowing dreamers to physically act out their dreams. RBD typically affects older adults, predominantly men, and involves dream enacting behaviors that can be vigorous and violent. People with RBD might punch, kick, leap out of bed, or engage in complex movements, all while remaining asleep or in a semi-conscious state. These behaviors often lead to injury to themselves or their bed partners, sometimes serious injuries. What makes RBD particularly concerning is its association with neurodegenerative diseases, particularly Parkinson's disease, Lewy body dementia, and multiple system atrophy, collectively called synucleinopathies because they involve abnormal accumulation of a protein called alpha-synuclein in the brain. RBD often appears years or even decades before other symptoms of these neurodegenerative diseases, potentially serving as an early warning sign. Not everyone with RBD will develop a neurodegenerative disease, but the risk is high enough, perhaps 80% over 10 to 15 years, that RBD is increasingly viewed as a harbinger of neurodegeneration. The mechanism of RBD involves damage to brainstem circuits that normally paralyze muscles during REM sleep. Animal studies have shown that lesions to specific brainstem regions, particularly the sublaterodorsal nucleus, produce RBD-like symptoms. In humans, RBD appears to result from the alpha-synuclein pathology affecting these same brainstem regions. Why the synucleinopathies first affect REM sleep control circuits before affecting other brain systems is not fully understood but is a subject of intense research interest. Treatment for RBD focuses on safety and symptom management. Environmental modifications, removing potentially dangerous objects from the bedroom, placing the mattress on the floor, using padded bed rails, help prevent injury. Medications, particularly clonazepam and melatonin, can reduce the frequency and severity of dream enacting behaviors, though they don't prevent the underlying neurodegenerative process. Ongoing research is exploring whether interventions that slow neurodegeneration might be beneficial if started early in RBD, before significant neurological damage has occurred. Beyond these more dramatic parasomnias, other sleep-related phenomena can cause distress or disruption. Sleep-related eating disorder involves eating during sleep with little or no memory of it the next day, sometimes leading to weight gain or consumption of inappropriate or dangerous substances. Sexsomnia involves sexual behaviors during sleep, which can be dangerous and lead to legal and relationship problems. Sleep talking is common and usually benign, but can occasionally reveal information the person would prefer to keep private. These various disorders and unusual experiences, nightmares, lucid dreams, sleep paralysis, sleepwalking, RBD, and others, reveal the complexity of sleep and the many ways it can go wrong. They also provide unique research opportunities. By studying what happens when normal sleep mechanisms fail, researchers gain insights into how those mechanisms normally work. RBD, for instance, taught us about the brainstem circuits controlling REM atonia. Sleep paralysis helped us understand the dissociation between consciousness and motor control during sleep-wake transitions. Lucid dreaming has shown that metacognitive awareness can coexist with REM sleep, challenging older views that all dreaming necessarily lacks self-awareness. For people experiencing these disorders, the message is that help is available. Modern sleep medicine recognizes these conditions as legitimate medical problems with established treatments. Sleep specialists can diagnose these disorders using overnight sleep studies, polysomnography, where brain waves, eye movements, muscle activity, breathing, and other physiological signals are monitored throughout the night. Accurate diagnosis often leads to effective treatment, whether through behavioral interventions, environmental modifications, medications, or some combination of approaches. Understanding when dreams and sleep go wrong also underscores the importance of normal, healthy sleep. Most people's dreams, even the occasionally weird or mildly disturbing ones, represent the brain doing its job, processing information, regulating emotions, and maintaining psychological health. When sleep is chronically disrupted, whether by lifestyle choices, environmental factors, or sleep disorders, the consequences extend far beyond mere tiredness. Sleep disruption affects memory, emotional regulation, decision-making, immune function, metabolic health, and overall well-being. Protecting sleep, ensuring adequate duration and quality, becomes one of the most important things we can do for our health. And part of that healthy sleep is dreaming, those strange nightly journeys that, when all goes well, contribute crucially to making us who we are. The future of dream science. As we reach the final stretches of this journey through the science of dreaming, it's worth pausing to appreciate how far we've come and to peer forward into what the future might hold. Less than a century has passed since Aserinsky first observed rapid eye movements in sleeping subjects. Yet in that time, our understanding of sleep and dreams has been utterly transformed. We've progressed from viewing sleep as a passive, unconscious state to recognizing it as an active, complex process involving multiple stages, distinct neurochemical states, and crucial functions for memory, emotion, and cognition. We've moved from philosophical speculation about dreams to empirical investigation using increasingly sophisticated tools. And we're now at a point where science can not only describe dreams but is beginning to influence, modify, and even decode them. The technological advances driving dream research forward are remarkable. Brain imaging techniques continue to improve, offering ever higher spatial and temporal resolution. Functional magnetic resonance imaging, or fMRI, can now identify patterns of brain activity associated with specific dream content. Researchers have shown, for instance, that they can predict with better than chance accuracy what broad category of content a person is dreaming about, visual scenes, objects, faces, based solely on their brain activity patterns during sleep. This represents a primitive form of dream decoding, reading out aspects of subjective experience from objective neural signals. Even more sophisticated approaches are emerging. Machine learning algorithms trained on large datasets of brain activity and dream reports are beginning to identify the neural signatures of specific dream elements. Some researchers have proposed that in the not-too-distant future, we might be able to create crude reconstructions of dream content, not reading minds exactly but inferring general themes and content from brain activity. This raises fascinating questions about privacy, consent, and the nature of consciousness itself. If dreams can be decoded, should they be? Who owns the content of dreams? These are questions our society will need to grapple with as the technology advances. Alongside decoding dreams, researchers are exploring methods for influencing them. Targeted memory reactivation, a technique where specific sensory cues associated with learned information are presented during sleep, has been shown to enhance memory consolidation for the cued information. If you learn pairs of images and sounds while awake and then hear just the sounds during subsequent sleep, your memory for the images associated with those sounds improves. This technique is being explored as a potential tool for enhancing learning, potentially even for treating memory disorders. Could targeted memory reactivation be used to influence dream content more broadly? Preliminary evidence suggests yes. Presenting odors, sounds, or other stimuli during sleep can influence what people dream about without necessarily waking them. This opens possibilities both intriguing and unsettling. Might people use this to enhance pleasant dreams, reduce nightmares, or process specific emotional experiences more effectively? Might advertisers someday attempt to influence dreams? A prospect that sounds more like dystopian fiction than science, but isn't entirely implausible given commercial interests in influencing consumer behavior. Another frontier involves lucid dreaming and the development of technologies to induce or enhance lucidity. Lucid dreaming has potential applications from nightmare treatment to skill learning to simply making sleep more interesting and controllable. Various approaches are being explored, from transcranial electrical stimulation delivering weak currents to frontal brain regions to increase prefrontal activity during REM sleep, potentially promoting lucidity, to devices that detect REM sleep and deliver sensory cues designed to trigger lucid awareness without causing awakening. The success rates of these techniques vary, and none reliably produces lucid dreams in all users. But the rapid pace of technological development suggests that more effective lucid dreaming induction methods are on the horizon. This raises interesting questions about whether everyone would want more lucid dreams. Some people find them fascinating and enriching. Others might prefer the uncontrolled, surprising nature of ordinary dreams. As with many technological developments, lucid dreaming technology might offer choices, allowing people to select how they want to experience their sleep that weren't previously available. Dream science is also contributing to our understanding of consciousness more broadly. Dreams represent a naturally occurring altered state of consciousness, one we all experience nightly. By studying how brain activity during sleep gives rise to conscious experience, how sensory disconnection from the environment affects perception and cognition, and how different neurochemical states create qualitatively different conscious experiences, researchers are building theories of consciousness that extend beyond just explaining waking awareness. Some theories, particularly integrated information theory and global workspace theory, make specific predictions about conscious experience during different sleep stages, predictions that can be tested through careful experimentation. The relationship between sleep, dreams, and mental health continues to be an area of intense research interest. As we've discussed, nearly all psychiatric disorders involve sleep disturbances, and many involve specifically altered dreaming. Depression typically involves early morning awakening and changes in REM sleep. Anxiety disorders affect sleep onset and maintenance. Psychotic disorders can involve vivid, disturbing dreams and altered dream content. Understanding these relationships better might lead to improved treatments. Already, interventions targeting sleep and dreams are showing promise for treating psychiatric conditions. Cognitive behavioral therapy for insomnia has been found to reduce depression symptoms, even in people whose primary problem is depression rather than insomnia. SE imagery rehearsal therapy for nightmares can reduce PTSD symptoms. Medications that normalize REM sleep can improve mood in depression. As our understanding of the specific sleep and dream abnormalities associated with different psychiatric conditions becomes more refined, we may be able to develop targeted interventions that address these abnormalities, potentially offering new treatment approaches. Artificial intelligence and machine learning are also transforming dream research. Large-scale analyses of dream reports using natural language processing can identify patterns and themes that would be impossible to detect through manual analysis. Machine learning models can predict dream content from waking experiences, personality traits, or even genetic markers, helping us understand what determines the specific content of individual dreams. These tools are also being applied to the massive datasets generated by sleep studies, identifying subtypes of sleep disorders, predicting which patients will respond to which treatments, and uncovering relationships between sleep patterns and health outcomes that weren't apparent through traditional analysis methods. One particularly exciting direction involves the intersection of dream research with evolutionary biology and comparative neuroscience. By studying sleep and dreaming across species, from simple invertebrates to complex mammals, researchers are tracing the evolutionary origins of sleep and beginning to understand why different species have such varied sleep patterns. Why do dolphins sleep with one brain hemisphere at a time? Why do some birds sleep while flying? Why do humans need so much REM sleep compared to other primates? These comparative questions can illuminate the functions of sleep and dreams, revealing which aspects are fundamental to nervous systems generally and which are specific to particular ecological niches or cognitive demands. There's also growing interest in individual differences in dreaming. Why do some people dream in vivid color while others dream in black and white? Why do some people frequently remember their dreams while others rarely do? Why are some people prone to nightmares or lucid dreams? Genetic studies, personality research, and brain imaging studies of individual differences are beginning to answer these questions, revealing that dream characteristics have some genetic components, are influenced by personality traits like imagination and anxiety, and correlate with structural and functional differences in brain organization. Understanding individual differences might allow for more personalized approaches to sleep medicine and mental health treatment. As we look toward the future, several major questions remain tantalizingly open. The most fundamental is also the simplest to state. Why do we dream? Despite decades of research and multiple theories, we still don't have a complete, universally accepted answer. Dreams likely serve multiple functions: memory consolidation, emotional regulation, threat simulation, prevention of neural overfitting, and perhaps others we haven't yet identified. Future research will need to integrate these different perspectives into a more comprehensive understanding. Another major question involves consciousness itself. How does neural activity give rise to subjective experience? Why is there something it's like to dream, rather than the brain just processing information unconsciously during sleep? These questions about the hard problem of consciousness might find unexpected answers through dream research. If we can understand how the sleeping brain generates vivid, complex conscious experiences in the absence of sensory input, we'll have made significant progress toward understanding consciousness in general. The relationship between dreams and creativity also deserves more systematic investigation. Anecdotal reports of creative insights emerging from dreams are numerous, from scientific discoveries to artistic inspiration. But rigorous empirical research on dreams and creativity is limited. Can we predict who will have creative dreams? Can we enhance creativity through dream manipulation? Do different types of dreams contribute differently to creative thinking? These questions have both theoretical interest and practical implications for education, problem solving, and innovation. Finally, as technology increasingly pervades every aspect of life, including sleep, we need to carefully consider the ethical implications. Should we develop technologies that allow dream decoding? Should we allow companies to influence dreams through targeted sensory stimulation? Should we make lucid dreaming easily accessible to everyone? Or might there be risks we haven't yet identified? Should dream content be considered private thought, protected from surveillance and intrusion? These aren't just abstract ethical questions, but practical issues that our society will need to address as dream technologies become more powerful and widely available. The science of dreaming stands at a fascinating juncture. We know enough to recognize dreams as important, meaningful experiences arising from specific neurobiological processes and serving crucial functions for mental and emotional health. But we still have much to learn about the details of those processes and functions, about individual and species differences in dreaming, and about how we might harness this knowledge to improve human well-being. The journey that began with Aserinsky's observation of his sleeping son continues, driven now by sophisticated technology, interdisciplinary collaboration, and a deep curiosity about one of the most universal, yet still mysterious aspects of human existence. As you drift towards sleep tonight, perhaps you'll think about the incredible journey your brain is about to undertake. The electrical rhythms will slow, neurons will fall into synchronized patterns, and neurotransmitters will shift in their delicate balance. Your consciousness will fade, sensory connection to the outside world will diminish, and somewhere around 90 minutes into sleep, your first REM period will begin. Your eyes will move rapidly beneath closed lids. Your muscles will become temporarily paralyzed. Acetylcholine will flood your brain, and you will dream. You might not remember those dreams tomorrow morning. Most people don't remember most of their dreams, and that forgetting isn't a failure but likely part of the process, keeping dream memories from cluttering waking consciousness. But whether you remember them or not, your dreams will have done their work. Memories will have been replayed and consolidated, strengthening some connections, weakening others, integrating new experiences into the vast network of everything you've ever learned. Emotions will have been processed, their intensity modulated, their content connected to broader patterns and past experiences. Your brain will have maintained its flexibility, explored novel combinations, prevented the overfitting that would make you rigid and unable to adapt. You will have run through scenarios, some threatening, some rewarding, exercising neural circuits and perhaps preparing for challenges you might face. And you will have experienced something remarkable, something uniquely human, yet something we share with cats and dogs and whales and birds. You will have visited places that don't exist, interacted with people who might be composites of many people or pure inventions, experienced events that never happened but that felt completely real as they unfolded. You will have been the artist and the audience of a private performance, a show that exists only for you, created by you, about you, serving purposes you're only beginning to consciously understand. The ancient peoples who wrote dream books and built dream temples, the philosophers who pondered the nature of dream experience, the psychoanalysts who sought hidden meanings in dream symbols, they were all grasping towards something true. Dreams do matter. They're not divine messages or prophetic visions, but they're not meaningless noise either. They're the conscious surface of crucial brain processes, the subjective experience of the night shift when your brain processes, integrates, and makes sense of your life. Modern science has revealed the mechanisms, the neurons and neurotransmitters, the brain regions and their interactions, the memory replay and emotional processing. But knowing the mechanisms doesn't diminish the mystery or the beauty. If anything, understanding how brains create dreams makes them even more wonderful, a testament to the extraordinary capabilities of 3 pounds of biological tissue, the most complex structure we know of in the universe, creating every night rich, vivid worlds from nothing more than its own activity patterns. So tonight, as your eyes grow heavy and consciousness begins its nightly fade, remember that you're not just shutting down or turning off. You're transitioning to a different mode of being, one where the brain's work continues, where memories are strengthened, emotions are processed, and flexibility is maintained. You're about to dream, and in doing so, you'll participate in one of the most remarkable phenomena in all of nature, one that scientists have only begun to understand, one that makes you uniquely you, one that has fascinated humanity since the first person woke and wondered what those strange nighttime visions meant. Sweet dreams. Your brain has important work to do. If you found this journey through dream science fascinating, please consider liking this video and subscribing to Deep Science Sleep. We explore the hidden mechanisms behind everyday phenomena, bringing you the latest scientific discoveries in a format perfect for learning while you drift off. Until next time, sleep well and dream deeply.